Gene expression. In the same year Watson and Crick published their model for the structure of DNA, Frederick Sanger at the lab of the same university showed that amino acids were the building blocks of proteins. Humans require 20 different amino acids. Some are made by the normal metabolic processes of the body, and some can only be derived from the diet. An amino acid is connected to other amino acids by a peptide bond. Two amino acids connected together is called a dipeptide. Three amino acids, a tripeptide, and any more than that is just simply a polypeptide. The chemical nature of amino acids causes them to attract or repel each other, so a particular combination of amino acids will result in a specific shape. The resulting polypeptide, or protein, is then able to influence the structure, function, or development of the cell. It didn't take long for the association between the sequence of amino acids in a protein and the sequence of nucleotides in a piece of DNA to be established. The specific sequence of nucleotides forms a specific sequence of amino acids, which conform to a specific protein. The sequence of nucleotides that indeed result in a protein is called a gene. Gene expression, then, is the protein that results from a gene. All the DNA is found in the nucleus, but the protein-making hardware is found outside in the cytoplasm. How does the genetic code move between the nucleus and the cytoplasm? RNA. RNA takes only the nucleotide sequence of the gene in a process called transcription, and converts the code once in the cytoplasm to a protein in a process called translation. This is referred to as the central dogma. Genetic information passes from the gene on the DNA to the RNA, then the RNA directs the assembly of a chain of amino acids to produce a protein. So during transcription, the gene on a piece of DNA is copied onto messenger RNA. It is important to note that gene expression is part of a cell's normal metabolic activity and therefore occurs during interphase of the cell cycle. Transcription will only begin if there's a need for that particular protein. So not all genes are expressed in all cells all the time. Quite the contrary, in fact. But if a protein is needed, it begins with RNA polymerase binding at the promoter region near the start of the gene of the DNA. This region is usually recognized by the RNA polymerase by a series of repeating adenine and thymine nucleotides. The DNA molecule opens up at that position. Doing so reveals two rows of exposed nucleotides. The lower row, say, is the strand the RNA nucleotides bind to. We call this the template strand. RNA nucleotides, like DNA nucleotides, are found floating around the nucleus. RNA polymerase moves along the DNA, attaching complement RNA molecules to the template strand of the DNA. The McGraw-Hill text doesn't call this strand the template strand, they call it the sense strand. There is absolutely no consensus regarding which strand is the sense strand. For every resource that states the sense strand is the template strand, there is a resource that says the sense strand is the coding strand. The template strand is the strand RNA nucleotides attach to, and has the same base sequence as the coding strand of the DNA, except uracil is replaced by thymine. Although I've seen diploma exam questions even refer to the coding strand as the template strand. It's best not to use the sense-antisense notation altogether. If a question appears on a diploma exam that asks you to translate a sequence of nucleotides, always assume it's talking about the template strand, no matter what name the question is calling it. In this case, the template strand is the lower strand. Notice that the coding strand, the upper strand, has the same sequence of nucleotides as the growing RNA strand. Since the RNA strand, once detached from the DNA, is now carrying the genetic message for that particular gene, we refer to this RNA strand as messenger RNA or mRNA. So at the end of the gene, the mRNA strand detaches. The end of the gene is marked by a sequence of nucleotides that the RNA polymerase recognizes as the termination sequence. The RNA polymerase detaches from the DNA, the mRNA moves away, and the DNA molecule reconnects itself to resume its original conformation. During translation, the mRNA carries the code to the cytoplasm and converts it into a protein. The mRNA leaves the nucleus through pores in the nuclear membrane. Once in the cytoplasm, it will combine with the protein assembling hardware to make a protein. Ribosomes, structures in two subunits made from ribosomal RNA or rRNA and protein, attach to the RNA. As the mRNA feeds between the subunits, another RNA structure called transfer RNA or tRNA carries amino acids floating around in the cytoplasm to the mRNA ribosome complex. In studying this central dogma, it is essential that you try to use these links that are made available to you to see animations of this transcription translation process. Ribosomes bring together the mRNA strand and tRNA molecules carrying the amino acids. The model of a tRNA molecule shown here is carrying arginine amino acid, one of the 20 amino acids humans require. At the other end is an anticodon, the 3 nucleotide combination that complements the codon on the mRNA molecule. Specifically, the mRNA feeds between the subunits of the ribosome until it comes across an AUG codon. AUG is the initiator codon, and the anticodon is held by a tRNA molecule carrying a methionine amino acid. Therefore, all proteins start out having a methionine as the first amino acid. However, not all proteins actually have a methionine amino acid, which means some enzymatically driven process cleaves the methionine off some proteins. At the neighboring site on the ribosome, another mRNA codon is exposed. This time it happens to be a GUA, which is the codon that brings a tRNA molecule carrying the valine amino acid. Both sites have tRNA molecules with their respective amino acids. The first tRNA passes its amino acid to the second tRNA and leaves its binding site. The 2 amino acids are connected by a peptide bond. The ribosome moves forward, exposing new mRNA codon for a third tRNA. The cycle continues until the ribosome reaches the stop codon, one of several codons that don't connect any amino acid carrying tRNA molecules, but rather a release factor which occupies the vacant binding site of the ribosome, prompting protein synthesis to terminate, releasing the amino acid chain, which then twists and conforms to function as a specific protein. To match up the amino acids with an mRNA codon, this table is available to you as part of your data sheet in your diploma exam or your final exam. Here's a sample problem. What would be the sequence of amino acids produced by the following DNA template strand? Use your data sheet to answer this problem. Remember, if it was a diploma exam question, you would assume it was a template strand anyway. So this is the mRNA complementary sequence during transcription. And read from your data sheet, you would see that these amino acids align with each mRNA codon.